Here is a link of an activity for you to draw this graph by yourself. If you click on this link, then you will see an Excel file with the number, and if you draw that numbers, then there will be the graph coming up, such as this. That's an activity, and you can do it later by yourself, which is related to build an action spectrum. But what is action spectrum? Let's see in the next slide. The action spectrum is the rate of photosynthesis. What is the rate of photosynthesis? Because they can analyze and they can measure the rate of the photosynthesis based on the wavelength, and then they drew this graph that shows that the amount or the rate of photosynthesis, like in around 400 nanometer and 700 plus nanometer, are having the maximum photosynthesis rate. So that's why it is called action spectrum, which means that the photosynthesis is acting. So the action of the photosynthesis, that's why it's called action spectrum. The next slide is showing the absorption spectrum. This means that in which wavelength we had the maximum absorption, and if we see the maximum photosynthesis is related to the maximum wavelength, then they are showing together that the more energy will be absorbed, the more wavelength will be absorbed at a certain wavelength, then the more photosynthesis will happen. So that's the meaning of the absorption spectrum, which is almost similar to action spectrum. And in this area, with this wavelength, we have more absorption, and also we have more photosynthesis. And the same here, we have more absorption at this wavelength, and we have more photosynthesis. Okay, let's watch the video together.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: photosystem 2, cytochrome B6F, photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem 2 complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem 2, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. One photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem 2 are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem 1 complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
The ultimate source of energy for most life on Earth is the sun. Plants use the process of photosynthesis to capture the sun's energy and put it to work. Cells derive the energy for life by respiration, using compounds that were originally created by photosynthesis. Cellular respiration and photosynthesis are complementary processes. In photosynthesis, plants use carbon dioxide, water, and light energy to produce sugar and other complex molecules. Oxygen is released as a byproduct. The complementary process of cellular respiration converts oxygen and sugar into carbon dioxide and water. In the process, some energy is released to make ATP, and the rest is lost as heat. Photosynthesis can be broken down into 2 sets of reactions, light-dependent and light-independent. The light-dependent reactions transform the energy in sunlight to chemical energy in the form of electrons with high potential energy. These reactions result in the production of oxygen from water. The light-independent reactions use ATP and NADPH to reduce carbon dioxide from the atmosphere to sugar through a set of reactions called the Calvin cycle. The carbohydrates produced by the Calvin cycle are used in cellular respiration to make ATP for the cell. The light-dependent reactions of photosynthesis occur within the thylakoid membranes inside the chloroplast. The thylakoid membranes contain an array of proteins, as well as chlorophyll and other pigments. This complex, called a photosystem, absorbs light and initiates the reactions of photosynthesis. Light-dependent reactions have 2 photosystems. In photosystem 1, excited electrons are used to produce NADPH. In photosystem 2, they're used to produce ATP. A photosystem absorbs photons of light, which bump electrons from chlorophyll molecules to electron acceptors within the photosystems. the chlorophyll molecules are left oxidized. Photosystem 2 transfers the energized electrons to a molecule called plastoquinone via an electron transport chain. The transfer releases enough energy to transport protons across the thylakoid membrane. A proton gradient forms, with a higher concentration inside the thylakoid than outside. The proton gradient is important because it represents potential energy that the cell uses to produce ATP. Notice that photosystem 2 is now missing 2 electrons. How are they replenished? Photosystem 2 strips electrons from water, leaving behind protons and oxygen. Meanwhile, the electrons transferred to plastoquinone pass next to a cytochrome complex, and then to a molecule called plastocyanin. Photosystem 1 also absorbs photons, which boost electrons from chlorophyll molecules to an electron acceptor within the photosystem. The energized electrons are passed to a molecule called ferredoxin, then to an enzyme that passes 2 electrons and a proton to a molecule of NADP+. This reaction produces NADPH, an electron carrier that donates its energy to the light-independent reactions of photosynthesis. Notice that photosystem 1 is now oxidized. It has lost electrons. These electrons are replenished by electrons originating from photosystem 2. The result of these light-dependent reactions is the production of NADPH, a proton gradient that fuels ATP production and the release of oxygen gas into the environment. When electrons are transferred along the thylakoid membrane, they have differing energy levels. Arranged according to these levels, they make a Z pattern. Here, the photosystems are shown as arrays of chlorophyll molecules. The electrons transferred from water to chlorophyll are relatively low in energy. Photons boost them to a much higher energy level, where they're captured by the electron acceptor called pheophytin in photosystem 2. The electrons are then gradually stepped down in potential energy through a series of redox reactions, releasing energy that is used indirectly to make ATP. Light again energizes the electrons, which are ultimately captured in NADPH. In this system, light increases the energy level of electrons, allowing them to fuel the production of ATP and NADPH. One function of the electron transport chain of photosystem 2 is to pump protons into the interior of the thylakoid. The accumulated protons, or hydrogen ions, represent a form of potential energy. The enzyme ATP synthase taps this energy. The hydrogen ions have a tendency to flow down their electrochemical gradient. As they flow through a pore in the ATP enzyme, they make the enzyme spin. This conformational change in the enzyme drives the phosphorylation of ADP to make ATP. The chemiosmotic synthesis of ATP occurs in both chloroplasts and mitochondria. As we've seen, the light-dependent reactions occur in the thylakoid membranes and require light to produce NADPH and ATP. These molecules fuel the next series of reactions, which occur in the stroma. The light-independent reactions, also known as the Calvin cycle, can occur with or without light. The Calvin cycle consists of 3 phases: the fixation phase, the reduction phase, and the regeneration phase. In the fixation phase, carbon dioxide from the atmosphere combines with a 5-carbon molecule called ribulose bisphosphate, or RUBP. This process, in which carbon dioxide is made usable to life by incorporating it into an organic molecule, is called carbon fixation. The product of the initial reaction is an unstable 6-carbon compound that splits to yield 2 3-carbon molecules of 3-phosphoglycerate. The reduction phase begins when 3-phosphoglycerate is phosphorylated by ATP and concludes when it has been reduced by electrons from NADPH. The product is the phosphorylated sugar glyceraldehyde 3-phosphate, G3P. 2 G3P molecules are used to manufacture glucose. If the Calvin cycle didn't go through a regeneration phase, the chloroplast would run out of the RUBP molecules required to make glucose. To make a single molecule of glucose and to regenerate RUBP, the cycle must run 6 times. If 6 carbon dioxide molecules react with 6 RUBP molecules, the chloroplast will form 12 molecules of 3-phosphoglycerate and use 12 molecules each of ATP and NADPH. 12 G3P molecules will result. 2 of these will be used to make glucose, leaving 10 to regenerate RUBP. The regeneration of RUBP costs the cell additional ATP. Note that the carbon atoms balance. 6 5-carbon molecules have a total of 30 carbon atoms. 10 3-carbon molecules also have 30 carbon atoms. In addition to glucose, the G3P from the Calvin cycle can be used to make other organic molecules. The Calvin cycle allows plants to use carbon dioxide from the air to form the sugar molecules required by all organisms.